Good afternoon, everyone. Thanks for coming to Vital Learning. This is our last one for the fall 2013 semester, and we're already hard at work putting together a great schedule for the spring. In fact, I heard that the library wants to do a presentation on Google Glass, so that should be pretty cool. Keep your eyes out for that. Today we have Jay Park, who is our scientific computing specialist, Professor Kathy Van Hoelen, and Kevin Heath, and they're going to talk to us about their experiences using the Exceed supercomputing resource. Thank you.
All right, thank you. Welcome to Vital Learning today. What I'm going to talk about today is about Exceed supercomputer resources and a couple of use cases at Harvard College. So this is my agenda. So I'm going to introduce you Exceed and Campus Champion program and available resources to you. And I'm going to hand it over to, you're going to go first? Okay. Professor Kathy Van Hoelen for her experience on Exceed and her research project, and then I will give it to Kathy. So, what is Exceed? Exceed is a comprehensive set of advanced heterogeneous high-end digital services integrated into a general-purpose cyber infrastructure. So, this is next-generation general-purpose and NSF-funded infrastructure. This one, Exceed, is essentially a collection of services, including hardware, software, human resources, knowledge. So, Exceed components are first the coordination and management of the resources, hardware, software, and they do have this insertion service, technology insertion service. So if you have a simulation or infrastructure for them to try out, you can register your technology to them and they can try it out. Extended collaborative support service. Once you get this Exceed resources from Exceed, you can pair with other experts from Exceed and then work on your difficult problems closely. And also training, education, outreach service like this. So these are the Exceed service providers, nationally and internationally, including Germany, 17 of them. So they provide their hardware, software, and human resources for you. Best of all, it's free. And what is Campus Champions program? Well, the Campus Champions program supports campus representatives like me as a local source of the knowledge about the HPC resources and opportunities. It started in 2007. Purdue is leading it. And we joined in 2012. There are more than 150 member campuses nationally. And so what do we get as a Campus Champion institution? Exceed provide support for Campus Champion especially. Also provide training and updates to Campus Champions. And they, well, when you have a question as a user, well, actually, when I have a question for any Exceed resources, the response time is much, I would say, much faster for Campus Champions because we already set up some ways to communicate with them and efficiently. Also, we're given a lot of startup resources, like much more than startup allocations. So we're given that computing time. Also, we can add our users to under my account so that they can try the resources out. And there are forums and so on. And Campus Champion's role is to raise awareness locally. Like this, we also provide training and so on. So basically, Campus Champion is the local representative and liaison between Exceed and Harvard College. Joseph actually offered a little bit of my time for the other colleges, the Clement colleges, but it depends on my time. But you can ask me if you need any resources on Exceed. So, there are 154 as of November Campus Champions throughout the nation. I guess except Maine, all the states. So what is available to you? PC resources, especially hardware, there are four different kinds: HPC, HTC, visualization, and storage. HPC is high performance computing resources. HTC is high throughput computing. High performance computing resources, you care about how fast you can finish your job. High throughput computing, you need a lot of job to finish your, to get more results for your simulation or a better, better results from the more results. And there are visualizations results using GPUs. And also, they have a lot of storage resources. The software, certainly, and this extended collaborative support service is like you pair with other staff or researcher who is expert in your area to discuss more about your difficult problems and Campus Champions. So you can go to this URL. You can see a lot of different resources, HP system, visualization, and so on. So if I introduce you some of them, UT Austin, Texas Advanced Computing Center, they have Stampede. It's probably the fastest computer in Exceed resource pool. 7+ petaflop computer. That's a cluster system and has 205 terabyte memory, aggregated memory per year. And also, PAC has another petaflop machine called Lone Star. Are you using Lone Star? Yeah. That's one of the fastest computers in Exceed. It's also a cluster system, and it can also run serial jobs. So it's good for some part in serial jobs. And there's Kraken is also a petaflop machine. It's massively multi-processing system. So it has a different system configuration than the cluster. It's especially built for this massively multi-processing jobs. Also, Gordon in San Diego, it's a 341 teraflop machine. It's also a cluster. Well, this is especially for fast I/O. It uses flash-based I/O. So that's a pretty new machine. Also, Trestles is 100 teraflop machine, and Blacklight is our favorite SMT machine. SMT system is the shared memory machine. So wherever you log in, you got 32 bytes of memory for your program. When you get your queue started, you can use essentially half of them because it's divided in half. So you can use 16 terabytes at once. Also, there are high throughput computing tool. Also, visualization resources. They have a lot of storage system, but it's kind of tricky because this is all project-based allocation. So once your project is done, probably you need to extend your years, or you will need to move it to another location. But anyway, that's available. And softwares. There's a lot of software available, and you can search for it. For example, if you need MATLAB, they have MATLAB license. You can search MATLAB, and it will show the resources that has MATLAB installed. Gaussian also. There are several resources that are Gaussian. They can install your software actually. If you have license, and you can ask them to install it for your group. Who uses XSEED? Physics, biosciences, astronomy, atmosphere, sciences, and chemistry, and natural systems. Over 32 fields of science. And 2 billion SUs is essentially 1 core hour on those machines. Over 2 billion core hours in those. So how can I get started? So you have 3 kinds of resource types: startup, education, and research. Startup is approved very fast, within 2 weeks. So within 3 or 4 weeks, you can start using those resources once you submit your proposal. It's not actually a long proposal. You just need to send them as your CV and 1-page abstract, less than 1 page. And education resources, if you have a class using HPC system, if you don't have HPC system available right now, you can ask them to give you a certain amount of time on a resource for 1 year or 1 semester. Also, the research resources. Research resources, you don't have like a startup resource. You can get up to 200K core hours. But research allocation, there's no limitation, and it can be extended to multi-years. And besides that, you have campus champion allocation, so that you can pass your applications on different resources. So how can you get started? Contact me at CIS and let me know what you need on XSEED, what you do, what kind of computation you do, and you can start from there. And then you will create an XSEED portal account, and I will add you to my Campus Champions resources, and according to your needs. So Blacklight is something else. If you need a lot of memory, I will put you in on the Blacklight users and so on. And you test it. Once you feel okay to use XSEED and you want to use XSEED resources for your research for a year, you can now apply for the startup allocation. So this is my, not mine, helpdesk@hmc.edu email. And they will forward it to me, or you can email me directly, jehu_park@hmc.edu. Also, you can email directly to XSEED. So, how is it now? Exactly 15 minutes. So I will hand it to Professor Van Heuvel.
I've been using XSEED to run some computational chemistry projects. But before I get started telling you about the details, I really owe a large thank you to Jay Cook, who was very instrumental in getting this going for me. everywhere from telling me that Exceed exists and that I could apply, to letting me use his Campus Champion allotment with my students this summer to see how well it worked for us. He ran personalized training sessions for me and my students to make sure we could actually get things going in a timely way. He helped me with my startup proposal, and he's been very patient with all of my sometimes phone-headed technical questions along the way as well. So thank you very much. The project that I've been doing is in silico investigations of biologically inspired catalysts. So computational chemistry is not always familiar to everyone. So I'll start by giving you a brief introduction to the field, the kinds of questions we want to answer, the particular project I'm doing, and then the results that we gained from Exceed. So the Nobel Prize in Chemistry this year went for advances in computational chemistry. And I love this picture that the Nobel Committee put out in their press release because it shows a French scientist working closely with a computational scientist, one informing the other. Chemistry remains an experimental science, but as advances in computers have made computational work much more feasible, there's much more give and take between experimental data, making predictions, or computations, putting them together. So the kinds of questions you can look at, you say a molecular geometry. Within a molecule, what are the bond lengths? What are the bond angles? This can be really important if you're talking about something like a large enzyme that has a particular pocket for a molecule or a substrate to bind. So you can see how well does the substrate actually fit in there? How likely is this reaction to go? You can also use computational chemistry to model spectroscopic properties. So spectroscopy is the study of how light interacts with matter. And there's a whole host of spectroscopic techniques that are commonly used in the lab. So in an example, this is a paper that came out last year where the scientists are modeling how the atoms in this molecule, 2,5-pyridine, are vibrating. Experimentally in the lab, they would vibrate in such a way to give you the black spectrum here. Then the scientists in this paper used computational methods to calculate or to predict on this vibrational spectrum. They got one result with the blue trace shown here. They added in a new approximation and got a different trace, a red trace. So I think what this really highlights is that depending on the theoretical treatment you use, computational chemistry will give you different results. So it's very important then to use your computations in conjunction with experiment to make sure what you're putting in the computer gives you something physically reasonable coming out. Then the last type of question that I'll talk about would be how does a chemical reaction actually work? So in a chemical reaction, two chemicals come together, they react, and you get something new. This comes at a price. There's an activation energy you need to put into the system to make the reaction go. The more activation energy you have, the harder the reaction is to get started. There are new computational methods where you can calculate how big is this activation barrier. If I use molecule A, what activation barrier will I get? If I change my system to molecule B, does the activation barrier get bigger or smaller? That could inform which one you want to make in the lab. So these are the kinds of questions that we're looking to answer today. We use computations in conjunction with experiments. So my students make molecules in the lab. They study how they react. They study them spectroscopically. Also use computations with give and take among all three, so that each one will give us a different piece to look at our system as a whole. So I work in the field of bioinorganic chemistry, which I think is best explained by looking at the label of a vitamin supplement. So you see vitamin A, vitamin C, vitamin D, the usual ones we hear about in health news. But you also see zinc, copper, manganese, chromium, molybdenum. Why are these in my vitamin? I've highlighted vitamin B12 as well because that contains cobalt. And in fact, the cyanocobalamin within a vitamin tablet has cobalt bound to a small cyanide molecule. But that's okay because molybdenum is in an enzyme that takes cyanide and converts it to something less harmful in biological systems. But the questions that my field asks then is how do we use these transition metals? What role do they play in nature? And increasingly, people are saying, wait a minute, These elements are abundant, they're inexpensive, and they're doing all kinds of remarkable transformations, chemical transformations under benign conditions. So can we understand how they work in, say, an enzyme, and apply that to design a better system to do that same chemistry in industry or in the lab using cheap materials, mild conditions? The two particular reactions my lab is looking at, the first one concerns methane, shown here. This is the main component of natural gas. It's a very energy-rich molecule, but methane gas is very diffuse. It's a flammable gas that can be hard to transport and store. What people really want to do is take methane and turn it into methanol, which is a liquid, much higher density, so the same volume of methanol has much higher energy density than the volume of methane. Well, this kind of chemical transformation, which is highly selective, can be done in nature by an enzyme that uses nickel in its active site. This was a pretty recent discovery, and it's not clear at all how this kind of novel reaction pathway works. We'd like to understand how to manipulate methane in this way. The second type of reaction that we're looking at is also somewhat in the field of also uses this enzyme. And the second reaction we're looking at is the same enzyme can take something like carcinogenic pollutant. This is trichloroethylene. It's been found in groundwater, it's carcinogenic, but enzymes that contain nickel and cobalt can take this pollutant and transform it into something much less noxious. We're interested in this because each of these red dots here indicates a well in the U.S. where one of these carcinogenic pollutants has been found. And in fact, yesterday I was looking at the newspaper from the Twin Cities, my old hometown, and one of the headlines was testing shows a high level of these pollutants in a residential neighborhood. So it's important to be able to think, how can we take it and deal with these pollutants? So the same enzyme that we were using in our methane studies can do this reaction. Also vitamin B12, which contains cobalt. So essentially the questions we're asking is, if we make model compounds of these rather complicated enzyme active sites, will the models reproduce the reactivity we want? Can we understand how the reactions work? And can we use these kind of fundamental insights to design a better catalyst to do these reactions? So I came to Harvard Med with a pretty big library of possible model compounds to study. And my students have been extraordinary because they've actually made a lot of these much more quickly than I expected. But what we want to start to do is think about not just making them in the lab, but we wanted to study them computationally as well. And so over the summer, my students began computational studies of all these different compounds. I also want to note, these are pretty big systems. In computational chemistry, the more atoms you put in, the longer your job takes to run. These compounds also use transition metals, which have lots of electrons. They're very hard to model using quantum mechanics in any kind of computationally cheap way. So the jobs can take a little longer to run because of the not just the number of atoms, but the type of atoms involved. So the way our workflow goes is you take experimental data of some sort to figure out what the molecule might look like. The students use a program called Gauss-View, which is available. Harvard Med has a license. It uses to visualize the molecules and create an input file, which they uploaded to Exceed. Then they took the results, the output file, compared it to an experiment, and we continued until we had a satisfactory model of the systems. So this would be an example of some of the data the students were able to extract. This was work by Casey Cannon, where he looked at various bond lengths in this cobalt compound and found what would be the lowest energy geometry. He did this for a whole host of compounds. I won't talk about the results, but he was able to make small perturbations to his system and see how the relative bond lengths change as a result. And then my students, Philip Woods and Jennifer Rogers, did something very similar with a different set of compounds. What I'm showing here is a comparison of the structural parameters of their model compounds as compared to the enzymes of comparative computation to experiment to see what might be the best model. And the way they set up their table, I thought was very nicely color-coded it, such that all the numbers in yellow are comparable bond lengths, those of green match up, and so on and so forth. So you can start to see what models might be the best ones to use. So the main advantages we got from Exceed this summer was it was very easy to use. We were all able to get started right away, which was really valuable. The computational times are also extraordinary. When I ran these kinds of computations in grad school on just a small cluster that we had in the chemistry department, they would take anywhere from 3 days to a week to run. With Exceed, we're seeing times of 3 to 9 hours. So we can get through far more calculations, and it's less wait time for the students. Along those same lines, we can apply higher levels of theory now to our systems and get more accurate results. And this was key too. Multiple students can submit multiple jobs to the queue all at once. We have another piece of software where I only have it on 1 or 2 PCs in the lab. It takes about a week to run. And so there's some impatience there. You know, when I tell the students, I'm so sorry, but so-and-so's job is still ready. We'll have to wait for 2 years. This, we were able to avoid that. And I also used Exceed this fall as part of an outreach program where I worked with the Scripps Academy math and science scholars in the program. And with a team of 4 high school girls, we made computational models of vitamin B12, submitted them to Exceed, and then looked at the structural parameters to see what best reproduced the natural system. Exceed is very valuable here because the girls were quite prolific in the models that they generated. Some of their models were rather imaginative, and the farther away you start from reality, the longer your jobs take. So I was allowed to have Exceed for that as well. There's a lot of people who think J-Hope. Robert Cade, who is a computational chemist here, is very gracious in letting me use his cluster while I was getting things going on Exceed. My students are doing remarkable. Paige Allen and Jessica Inamoto did synthetic work. Philip Woods, Casey Cannon, and Jennifer Rogers did both computations and synthesis. And Judge Lee has been working with me this fall to do benchmarking studies. Funding came from the chemistry department, the Stafford Foundation, and the Center for Environmental Studies. And I'm happy to answer any questions that you have at the end of this presentation.
So, thank you very much.
So I would also like to start off by thanking J-Hope, who was very, very helpful. He introduced me to Exceed, he showed me around what it is, how to get set up with it, and actually get stuff running. And it was fantastic because instead of having to look through all of the resources, which are very nice, J-Hope was able to help me and work with me and get things up running very quickly, which is really helpful for my research. So my research project this summer was to design a general parameter estimation algorithm for biological models. Well, what does that mean? When you're designing biological models, what you're trying to do is find a quantitative way to express something that you notice in biology. Maybe it's a signaling pathway, maybe it's how your hair grows. But no matter what you're doing, you design variables and you try to give them values. And the more accurate those values are, the closer your model represents reality. Now, the problem with this is that it can take a lot of time to figure out what those values actually are. If it's a linear model, you can guess somewhat easily. You can just go through and say, what if I try this value and these sets of values? But when you have nonlinear models, you aren't guaranteed a solution. You can guess as much as you want, but you aren't guaranteed to actually find something that represents reality well. Those values may be out there, but the trouble is, how do you actually find those? So the answer is, instead of just brute force trying to calculate those values, we do parameter estimation. We use computers to guess what those parameters might actually be, what those variables should be. So what we end up doing is trying to find the optimal values, and we create a function that says when these values equal some number, then this is an optimal representation of reality, and we can use the computer to actually calculate what those values are. So what are some nonlinear or low optimization methods of parameter estimation? First, there's stochastic methods. This is essentially random guessing. Rather than going through every possible value, you guess at different values and see, is this a better representation of reality or is it worse? And we can just keep on doing that as much as we want until we get a satisfactory answer. Next, there's clustering, which is where you take global optimization methods, linear methods, and you apply that to different points on your optimization curve. So you say, I have this linear slope sort of on my curve, and I'm going to start at the top, and I'm going to go down until I find the best values possible. Then there's evolutionary strategies. You sort of model evolution, create populations. You have organisms that represent your parameters, and you start off with some initial value. It doesn't really matter what it is, and then you mutate it. You change them. Say, increase some parameters, increase others, and over time, eventually, you'll get a better answer. Then finally, there's simulated annealing, which is the most different form of test, you could say. It sort of comes from a physics standpoint. It's simulating the cooling of a metal actually. What happens is, when you're cooling a metal, at first you start off just rapidly cooling things down. It's very hot, so of course it disperses the heat very quickly. But as the metal gets cooler, the rate of cooling slows, and this allows metal to take on a more uniform structure. So you can get a crystalline structure in the metal rather than sort of a haphazard lattice work of crystals that don't necessarily all work together. And the goal of simulated annealing is to slow down the rate of change in your parameter estimation, so you can find a better answer as you get closer and closer to your goal. So how does this all work in general? So the algorithm I designed has three main components. The first major component I call optimized island, where island is sort of a population of organisms. So what you do is, what I did was I created a population of organisms. Each organism represents a parameter in our biological model. And then I perform natural selection on the organisms, on the populations I find the best ones. And then I reproduce those populations, those organisms, and create more of them. And I check them, and I keep on changing them until I get a satisfactory population. And the wonderful thing about this is that I can do this with a whole bunch of different populations, a whole bunch of different islands. I can keep on doing this technically as many as I want. And the more I have, the more accurate my answers will be. Next, talk a little bit about reproduction. First, we calculate the mutation for each organism. Then we create new organisms based off of your current organisms. We mutate them correctly to some value can either increase or decrease. We don't necessarily know who's going to be better, but hopefully it is actually better. Then you sort of compare them and you get rid of the ones that you don't want. It's a brutal process, but it's very efficient. Basically killing off what you don't want, killing off is worse. And finally, mutation. I won't go into that much detail, but a large part of my project was actually figuring out how to properly so that you can have results than just random changes.
So you parallelize the optimizable island.
Yes. So the reason I did it this way is that optimized island can be parallelized. You can create multiple populations. You can have multiple islands of these populations. And you can do all of them at the same time. Parallelized. In Serial, what you normally do is you start with an island and you create a population. You go through and create all the organisms. And you go change all the organisms. And then you go produce and you do that for each one sequence. However, if you can parallelize that, you can actually do that all at the same time. So you don't have to wait. So in order to test my algorithm, I created a fitness function. And it was a minimization fitness function. So you can see at the value where x equals -3, 0. And I designed it that way. So when all the parameters based on that function you see on the bottom equals 0, then that's the optimal solution. And this was designed with the optimal solution to test how efficient my algorithm was. And when you're actually trying to calculate the parameters of a model, you create a population and maybe organisms puts you there. So what happens is it mutates and then maybe it shifts down here. And then it mutates again and then it keeps on shifting down here and it finds, oh, this one's going to that. So this is a better solution relative to the others around. However, if you create more populations like a stochastic model, you can start with one over here. And it can mutate and keeps on going down and finds a better and better solution. What you can do is the more resources you have, the more computational power you have, the more populations you can create, the more times you can do this. And in the stochastic model, you can actually essentially brute force the answer. You have enough different possibilities that you will find the best solution. It's just a matter of not getting stuck in these troughs that are sort of decoy solutions. You actually want to find the good. So here's a sample run of my algorithm using parallelization. So I start off with just one thread. This is essentially serial. It took about 560 seconds. But as you can see, as you parallelize it, you increase the number of threads you're using to run. It becomes exponentially faster. So after about 8, with about 8 threads, which means I can do 8 different populations at once, it's about a 6-fold decrease in time. So 6 times faster. And you can see it does make a significant difference to be able to parallelize this. So finally, where does exceed go? So this is an example of a Q subscript, which will submit to exceed resource here and tell it to run reset. So this is a C shell. You can tell it how many CPUs you want to use, how long you want it to run for, what sort of job you want, where all your files are, and whether or not you want to parallelize it, and it'll do that all for you. This is fantastic because you can give it the script, you can give it the program, and you can let it run, and it will run it for you. And when it's done, it will tell you can see the results. So Exceed was very, very helpful for my research. Just because the computational time it takes to actually calculate the parameter models can become incredibly large. When you have just a basic model, like I showed you, that I was testing with, that was 6 different parameters that you can do fairly quickly. But when you end up with a biological model that can have over 50 parameters, and all those can vary between, say, -1,000 and 1,000, and you have so many different possibilities that you really need the computational research
very much. That's great. If I add a little bit more about these showcases, Professor Van Heuvelen's case was easy case because she just needed Gaussian software on those machines. So what we do at CISD was just to let them teach them how to use Exceed, how to log in, how to submit their job, and how to upload their files and so on. And Kevin's case was a little bit more harder. I started actually 6 months ago, last winter. Jacqueline, she came to me and talked about her algorithm and her serial algorithm, and then we started to parallelize it at that time, and in the summer, actually, these students could parallelize certain part of it so that they can run it on Blacklight. Actually, it was funny. I taught them how to run their program, and then after like a couple of days later, I got an email saying that my resources I only have 15% of resources on Blacklight. What? They've been using it for like running it like with hundreds threads, hundreds cores for certain hours, and then 1 core hour times hundreds is like having a lot of. But it's good because that resources for that matter. So it was funny, but it was a good experience. So I think that was it. Do you have any questions for us?
So I guess, because this seems like it's a system that's kind of global, run by a group, is there like, what separates this group of supercomputers from, let's just say, others? Like, why would someone want to pursue XC versus just another institution's supercomputer? Is it because you have a lot of resources at your hands?
So you have direct access to a supercomputer center?
Right.
The difference is that Exceed has their centralized system for service. It's actually comprehensive service.
Right.
So you get not only the hardware, but also the human support. If you happen to have any problem with the system, they will talk to you about it. So that's one of the benefits. And I don't know how you, well, I actually contacted San Diego Supercomputing Center for some resources. For example, San Diego Supercomputing Center, they only give out certain resources to California State University system researchers. Other than that, you need to pay for it. But this is all free for researchers.
I have a couple questions, if you don't mind, on the science a little bit. Why did you pick the genetic approach as opposed to, say, simulated annealing? I mean, you gave many methods of doing optimization, but then you just went off and said, I did this one. And I didn't know if there was a rationale for choosing that one versus the other.
Well, I did sort of simplify what my research was. In the end, I actually used stochastic, sharing, and simulated methods in my algorithm. So I sort of worked them all together. The overall framework of the algorithm was an evolutionary model, which is why I focused on that. But actually calculating the mutation rate was simulated.
Okay. Is there reasons for choosing? I'm just curious why you made those kinds of choices. Did you expect it to work better one way than the other, or was it just, let's try this?
It was more of, let's try this. I just wanted to see if I can get something better than just individual.
Okay. And my other question on that, Raman spectroscopy. So you had two simulations of different sophistications, I guess. But there was clearly one Raman line that was missed completely. right?
Right.
So how do you know when the model is good enough for your purposes? I mean, it clearly was not modeled in reality. Of course, it never does.
Right.
So how do you know when it's good enough for your purposes and when, that particular vibration you missed? Did you tinker?
Yes, so I think that's going to come down to looking at the origin of the one that you missed. So for example, is it a sample peak? Is it a solvent? You want to make sure it's from your sampler. You
Right.
try to get there. And I think clearly that was Raman spectroscopy is very hard to simulate because for that technique, you're not just looking technically at a vibration, you're looking at actual excitation and relaxation. So it becomes, whenever you model excited states, it becomes more complicated. So I think this was an advance in how Raman spectroscopy is modeled. Clearly, it's not perfect yet.
Another?
I saw you packed a cave and he has a cluster. How would your work have gone if you used his cluster? Would it be the same speed, slower, faster?
His cluster works well. It's going to be slower than Exceed. You just don't have the same kind of memory or processors and all of that. He was very kind in allowing us access, but that was during the sabbatical year. Now he's gearing up his own research program. And since the jobs I'm doing would take days or a week or so on his system, I wanted to make sure I wasn't imposing on a colleague. So I want to make sure we can. And he's very willing to help me. He would have given me time.
So what are the downsides of using
Downtime?
Downside.
Downside.
From a researcher point of view.
Maybe somewhat of a black box for the students. You send it off from here you can map it, but you don't physically get to see when it goes into the cluster. I'm not sure how it all.
Actually, I guess the way I approached it, I actually had it tell me all that data. So some of the commands I had in the shell script was actually to send me all of the data, what's going on, like how much time it's spent on certain things. And I can look at like the computational time where I got stuck, what is it that was turned on. You can also have it give you input on like the number of threads essentially using it different points in time. So I guess it sort of depends on how much you put into it. But it can be a black box or it can actually be. I feel like sort of like maybe the only downside I could think of is that depending on how many other scientists are using the supercomputer at the same time as you are, you might have to wait a bit. Usually for like smaller sample batches, like debugging, you don't really wait. Sometimes I would have to wait a while for my project to actually run, depending on how much time I want.
One of the downsides I see as a campus champion is that the start of a location is for only 1 year. And it's easy to get started, but after 1 year, the research is going on after that period. The researcher needs to submit rather involved proposal to get the actual research allocation. It's a multi-year allocation that is much more involved.
More involved than, say, an NSF grant?
Yeah, because it's NSF. Right? Once you get it, it's quite good resources for your research. Any other questions? All right, thank you.